Hello, and thanks for tuning in. In this talk, I'm going to share some of our recent work on Antarctica's only endemic insect, the Antarctic midge. This is part of a larger project that includes comparative genomics, population genetics, and comparative physiology to try to unlock the secrets that allow this amazing creature to survive further south than any other insect. And our lab is primarily responsible for the comparative physiology aspects of this project, and so I'll be sharing some of those results today. So when an organism faces a stressful environment, there are things that can happen before, during, and after the stress that can improve survival. Things that happen before include acclimation responses or more short-term hardening responses. And of course, there are dynamic stress responses that occur during a stress to help deal with some of the damage that's incurred. But then oftentimes overlooked is also what happens after the stress. And these include processes to repair injury and also allow that organism to resume normal development and continue its functions. And so today I'll be telling you about a little bit of each of these in the context of the Antarctic midge. And so in the first part, I'll deal with a concept called cross tolerance. So Antarctic insects, while we often think of temperature as the predominant feature of these environments, face many different types of stress. And so it's important to be able to deal with multiple stresses simultaneously. And in the last part, I'm going to talk a little bit about some recent work on long-term recovery from stress in the context of an energy budget model where there's a certain finite energy availability in these environments, and this energy must be allocated to important processes like growth and development, reproduction. And so stress responses, which also take energy, can detract from some of these other responses or other processes. So I'll look at this in the context of recovery from freezing. So for cross tolerance, to make a longer story short, I'll go through this quickly. If we take larvae and directly freeze them at minus 18 degrees Celsius, almost all of them die, which is shown here in the white bars. But if we give them a 2-hour mild stress treatment before the freezing, and this includes temperature and dehydration cues, various osmotic conditions, and also ultraviolet light, in almost every case, we see a dramatic increase in survival. The cases with stars are statistically significant. So this is showing that exposure to one type of stress can actually enhance the tolerance to a different type of stress. Now, to follow up on that, we were interested in, so when these larvae experience these different stressors, are there conserved or stress-specific gene expression modules that are important for coping with these stressful environments? And so in this study, grad student Jack Devlin took larvae, exposed them to either cold, heat, desiccation, hyperosmotic, or hypoosmotic stress for 24 hours, and these were equally stressful levels of these different stressors. And then the larvae were sampled either immediately after the stress or after 12 hours of recovery, and then used RNA-seq to analyze gene expression. And so we can test some specific hypotheses. There's more than this, but some of the ones that we're interested in are the hypothesis that stressors that are physiologically similar will also be similar at the molecular level. And we can also test hypotheses that based on physiological data, all the stressors will likely converge on core processes such as protein denaturation and oxidative stress. And that will be reflected at the molecular level. And also that we expect that metabolic processes are downregulated as the larvae shift their focus to stress recovery. And so what Jack did was, after all these this is showing results for immediately after stress. This is a very complicated plot called an upset plot, which shows the number of differentially expressed genes that are shared by different combinations of treatments. And so if there's a dot, that means that treatment is represented here. But importantly, if we look at all 5 stressful environments, only 5 genes are shared across all of those stressors, indicating that most of the changes in gene expression are specific to one type of stress. And then very quickly here, another grad student in my lab, Lima, is focused on recovery from stress. And this is looking at energy balance and gene expression during recovery from an acute freezing event, from a short-term freeze. Energetically, the larvae are just fine. Overall, over 15 days of recovery, there's no appreciable differences in energy balance between frozen larvae in blue and control larvae in gray. But at the gene expression level, we see there's a lot of things happening early on during recovery, within 1 day, few things happening later, but importantly, some of the genes that are still being differentially expressed later in recovery are involved in stress responses, indicating that there is some potential for long-term damage following a short-term freezing event. And so that's all the time I have. So I'd like to acknowledge our funding sources, as well as the wonderful staff of the Lawrence M. Gould and Palmer Station.